Everybody, welcome to chapter 5. This is the biological macromolecules chapter. Um, so, what we're going to talk about is how do the major uh, biological molecules that your body uses, how do they form? How do they break down? We'll discuss the four major categories of biological molecules and um, sort of their roles in different organisms' bodies. So, what do they do? Importantly, we're going to talk about proteins because proteins do a lot of the functions in our body. So we're going to really delve into the structure and try to understand how do proteins get made in your cells because the structure that forms determines what the function of that protein can be. So we'll talk about all of those things. So the basic way that most of the molecules in your body form is through monomer polymer chemistry. It sounds a little bit intimidating, but all that means is that you take a lot of very similar building blocks and you link them together into a long chain. Um, so if you think about a chain made out of metal, each one of those links is just a repeating unit again and again linked together. Well, each link is like a monomer, and then the whole chain is going to be your polymer. And that's basically how most of our biological molecules are formed. Now, when we add links to the chain, that's called dehydration synthesis. Um, and the reason it's called dehydration is because as you take two links and join them together, in order to free up uh, elements to bond together, you have to break off an H from one side and an OH from the other side. That frees up a covalent bond, which then will join the links. The H and the OH go together to make an H2O. So you're dehydrating the monomers, removing a water to synthesize a polymer. Um, and then just the opposite happens in a reaction called hydrolysis, or if you break it down, hydrolysis. Hydro means water. You add water to break apart. That's the lysis part. The word lysis means to break. So we're going to add an H2O, an H to one side, an OH to the other side, and you break your links apart into separate monomers. And here we can sort of see that reaction with the H and the OH, that forming H2O, freeing up these two covalent bonds to join together. And then here's the hydrolysis reaction, breaking that bond, um, adding a water, an H goes to one side and OH goes to the other. And more examples of that. Here we see two amino acids joining together. That's the OH and the H. That's where they come from. And down below here, these are all types of sugars. Again, an OH and an H are being removed, forming a water, making two sugars join together. So the first biological molecule that we're going to talk about are proteins. And proteins are arguably the most important biological molecule because they really do the work of the cell. Basically, since we started to understand the chemistry of life, we realized that a huge portion of your cells is made of proteins once you take the water out. So half of your cell's weight minus the water is made of protein. So we've always sort of known that proteins are really important. And we also knew that proteins are primarily made of the elements CHON and S. So that S is a little bit unique. Not many other biological molecules have sulfur in them. So that's kind of a distinguishing feature. And here's a picture of a fully formed myoglobin protein, similar to what we have in our blood. We call it hemoglobin. And we'll talk about how does this form come to be. So I've said that proteins do a lot of the stuff in our bodies. Um, these are all some of the various ways that proteins form and the functions that they can have. So the first one is enzymes. And enzymes are really important because they serve to speed up chemical reactions. That's their whole job is to come across a chemical reaction and make it happen much faster than it ever would without that enzyme being there. And we will do an entire lab on just enzymes. But proteins also are antibodies in our immune system. Some proteins are used as storage, like in milk, to transport to the young of the species. Um, some transport stuff around, like hemoglobin transports oxygen in our blood. Some are hormones that send signals, like insulin, to tell your body to do things. Some are receptors that receive messages. Um, some proteins are for movement. This is the one we really think of when we think of proteins, because what we eat is in the form of meat is mostly these motor proteins. And then structure, that's keratin, things like hair, skin, um, proteins that are tough and resilient and are able to defend us from the outside world. So a lot of functions in here. Here are some pictures of those things I just talked about. So, how do proteins have this really wide array of functions? And if you remember from chapter one, structure goes with function. So, in order to have all of these various functions, they have to have many different types of structures, and they do. So, what we're going to talk about now is how do we get from these simple monomers to much more complex polymers? And then how do the polymers get all these different shapes? So the monomer of amino acid or of a protein are amino acids. Here in this picture, you see this black carbon atom. Um, that's called the alpha carbon. Off of one side of that carbon is this nitrogen group, this blue ball. That's called an amino group. And off the other side, over here, you have another carbon double bonded to an oxygen, and that's an acid group. So we get the name amino acid from these two side groups. What varies from one amino acid to another is this little light blue ball here, denoted by the letter R. Um, that's not the element radon. What that means is it's a variable group, it's a random group. Um, and depending on what that group, that R group is, really defines what that amino acid, how it behaves chemically. And there are 20 different R groups that our bodies use to build all the proteins that we have. So that's sort of weird. There are only 20 different R groups. And when you think about it, you could attach just about anything to this carbon. Um, but there are basically only 20 variants of an amino acid that our bodies can recognize and use to build functional proteins. These are going to link together again, like links in a chain. Here we see a long chain of amino acids, and each one of these purple balls is a different amino acid. And if you look closely at it, there's three-letter abbreviations, and those abbreviations are what tell you what that amino acid is. So here's tryptophan, here's valine, here's phenylalanine. So there's just 20 different letters, and you can think of it as like spelling a word with a 20-letter alphabet. This particular chain of amino acids, you can see it's numbered. So it's about 27, I'm sorry, 127 amino acids long, which is actually a pretty small protein. So some of these chains can be really, really long. Again, here is the structure of a single amino acid. You have this central carbon. This nitrogen group is the amino. This COOH group is the acid group. You have a hydrogen, and then you have your variable R group up here. And here's a list of all 20 amino acids. Um, and what makes these different is the way the R group behaves chemically. So all of these in yellow are R groups that are nonpolar. They don't have any sort of charge. And if you look at them, it makes sense. A lot of carbons, a lot of hydrogens, none of those things are going to form hydrogen bonds. They're going to be nonpolar. They're not going to react with water, things like that. Here we have a sulfur, which is interesting. That one's sort of buried in between two carbons. Here we have a couple ring structures that are just carbon and hydrogens. Over here, there is an NH group, which does have a little tiny bit of reaction with water. And if anybody lifts weights or anything like that, you may have heard of the branch chain amino acids or BCAAs. A lot of drinks you can get now say with BCAAs. Well, these three, valine, leucine, isoleucine, those are your branch chain amino acids. And you can see the R groups have a branched chain. The reason that they market those types of supplements and beverages for people who lift weights, these are really important in building muscle. So you need a pretty much of those BCAAs, which you can easily get through the food you eat. You don't really need to take a supplement for it. Here in these green boxes, these are our polar side chains, so you see a lot of oxygens. If you think back to chapter 2 and 3, when you have OHs together, that's going to be a polar covalent bond, forming those little tiny partial positive and negative charges. So these are all going to interact with water. Here at the end, you have asparagine and glutamine. Those have NH2 groups, which have that slight charge. And then down here at the bottom, the acidic and basic amino acids, they have either a negative charge for the acidic ones or a positive charge for the basic ones. And those are ions. They're going to form ionic bonds with each other, things like that. So the amino acids we divide up by the chemistry of the R groups. Okay. And again, these are formed through a dehydration reaction. You lose an OH and an H, forming a new bond. That bond is called a peptide bond, specifically in building proteins. Okay. So the primary structure of a protein, the first level of structure, is the spelling. What order do those amino acids come in? So you have a 20-letter alphabet. What are those? What are the amino acids that it uses, and in what order? Just like spelling a word. The second level of protein structure, all of those amino acids start to interact with each other chemically. And they're going to form some very characteristic structures. Like here we have a helix called an alpha helix. And down here we have this sort of back and forth winding structure that forms a flat sheet. That's called a beta pleated sheet. And all these interactions at the second level of folding are happening because of these amino and carboxyl groups interacting. At the second level, the R groups haven't started reacting yet. So you're getting that helical winding shape and you're getting those flat sheets um, from what we call backbone interactions because these amino groups and these carboxyl groups start to do chemistry, start to interact, start to connect to each other. So first level, long string of amino acids. Second level, it starts to fold because of those backbone interactions. The third level of structure, now the R groups start to interact. So any of those polar ones are going to form hydrogen bonds. It's going to bend the chain of amino acids and it's going to hydrogen bond together. Over here, you can see these are some of the branch chain amino acids. They're going to form non-polar bonds together. Also, because your cells are full of water, they want to sort of turn to the inside of the protein to sort of hide from water, get away from it. So they're going to bond to each other in a response to moving away from water. Over here, we have an ionic bond forming between one of those acids and one of the bases. So that forms an ionic bond. And then the last type of bond that can happen are these disulfide bridges. So there's a couple amino acids that have sulfur in them. Sulfur really, really likes to bond to other sulfurs. So when two of these amino acids, these R groups, get close to each other, if they both have sulfur, they're going to actually form a covalent bond together. And that's a really strong bond. So as we look at these four, hydrogen bonds are going to be the weakest. Um, these hydrophobic actions are going to be weak. Ionic bonds are going to be pretty strong. And then these disulfide bridges are going to be really strong. So you have this sort of variability in, okay, a hydrogen bond formed, but it can break again because it's not a strong bond. And you might have one bond overriding other bonds. So a disulfide bridge, it's really strong. It may break some other hydrogen bonds in other places to form that really strong connection. So that's the third level of structure. It's the R group interactions. And then the fourth level, at the third level, let me back up, that chain is now fully folded, but it may not be a functional protein yet because a lot of proteins are multiple folded polypeptides together. So the fourth level of structure is when the protein gets its function and actually becomes a protein. So here we have three of these twisted chains. That's a collagen protein. It actually takes three polypeptides twisting together to make that. Here on the right, we have hemoglobin, which is four globin proteins or polypeptides, and then four heme polypeptides. And it's not until those eight pieces come together, four hemes, four globins, that it becomes a functional protein. And that is the fourth level of structure. So in your cells, these things, it doesn't go like, okay, step one, okay, step two, step three. These things all sort of happen at the same time simultaneously, but we break it down into these four steps to understand the overall process a little better. It's also possible that at the third level, this level, that's the finished protein because there are a lot of proteins that don't combine with other proteins. So enzymes, for example, might just be one big protein. Um, Once it's folded, it's done, and there is no fourth level because it was finished at the third level. So keep that in mind. These are not like hard and fast rules, one after the other. It's just a way for us to understand things. Um, Here are all four steps in order. Step one, long chain of amino acids. Step two, the backbone interacts. You form these helices, and flat sheets. The third level of structure, these are your R group interactions, polar, nonpolar, acids, bases, disulfide bridges, all of that stuff. And then here at the fourth level of structure, multiple subunits come together to make the final functional protein. There are some proteins that don't fold correctly. So they might get to that fourth level and they still don't really work that well. Um, that's where there are other proteins that step in that help them fold correctly. So this is an example of a chaperonin protein where the chain of amino acids actually goes inside of this other protein where there's special chemistry inside that forces the protein to fold in a very specific way that it normally wouldn't do. Um, so you can really think of these chaperonins like they're molds for clay. You push the clay in, and the clay is going to take the shape of the mold. That's what happens inside of a chaperonin, but it's more of a chemistry mold than anything. The other thing is, once a protein folds, it can still unfold. Really easy example of this is cracking an egg on a hot skillet. The clear part of the egg turns white. When that happens, what you're actually doing is taking a whole bunch of proteins. Egg white is basically pure protein, and those proteins, because of all that heat, start to unfold and they form a crystal structure. And that's what a cooked egg white is. It's a denatured protein, meaning that it's no longer in its natural form that it formed in. Tons of examples of this. Cooking steak, it turns brown. That transition from red to brown is denaturing. This can also happen if you expose proteins to things like strong acids. It can make them unfold and take a different shape. And once you unfold the protein, it is incredibly hard to refold it again. In a lot of cases, it's not possible. So you can't uncook an egg. Um, Examples of why shape and function are so important. So here is hemoglobin that we just sort of talked about. It's these four globin subunits. And in people who have the normal uh, hemoglobin gene or globin gene, you're going to make these globin subunits. It's going to form normally into these nice globular structures that carry oxygen. And this is going to fill up your red blood cells. So those red blood cells can pick up lots and lots of oxygen, travel through your circulatory system. One thing that a red blood cell has to do is get through capillaries. Capillaries are just big enough to allow red blood cells to get through one at a time. So capillaries are like single file for red blood cells. Because of these nice globin proteins, the red blood cells are very squishy and bendable. So as they're squeezing their way through the capillaries, they can bend and flex and go around corners and things like that. If you are a person who has a mutation on a gene, specifically here, the number 6 amino acid, it was a glutamine and now it's a valine. So there would be a mutation on the gene that would lead to this substitution. It just means that your globin proteins, some of them fold a little differently. So instead of being this sort of, I don't even know what shape that is, heart shape, whatever. They make this sort of letter F shape. Okay, that's going to form your 4 unit thing. But here's the catch. If you expose this mutated globin to low oxygen levels, so if you're a person with this at high elevations, if you're a person with this condition who is doing a sport, these globins actually link up and become this rod, basically this protein stiff rod. And your red blood cells take on this very rigid sort of crescent shape. And that is not flexible. So as these stiff crescent shaped cells go through your capillaries, they get stuck. As they get filtered, uh, as they go through your liver, things like that, they get stuck. As they go through your lungs, they get stuck. Um, and this is what sickle cell anemia is. So, this one simple mutation of the number 6 amino acid leads to this chain of events. Make the protein folds incorrectly. The protein then at the fourth level of structure makes these rod shapes. Those rod shapes change the way the cell is shaped, which then change the function of that cell and cause lots of problems with sickle cell anemia. So that's just one simple example. The good news is we all have mutations. Most of them aren't like this, that it causes severe problems. Okay. Um, So that's it for proteins. The next one is nucleic acids. We're not going to go into nucleic acids too much uh, in this chapter. Later on in the semester, we're going to get to chapter 16, where we spend an entire chapter just on this. So for this, all we need to know is there are two types of amino acids. One is DNA, one is RNA. DNA forms the double-stranded helix, and it stores your genetic material. Um, it spells out a code. That code is what tells your cells what proteins to build. So one gene written in DNA tells your cell how to build one protein. So we just saw globin. If you have a misspelling in the DNA, it can lead to an altered structure of a protein. RNA, on the other hand, it's a very, very similar chemical, um, but there's one slight difference in that there's an extra oxygen on it. That drastically alters the way it works and the way it behaves. RNA is single-stranded instead of a double strand, um, and it's one of its main functions in your cells is to transport the message. So DNA is the chemical, there's genes written on it. RNA makes a copy of that message and takes it to where the proteins are built. So RNA serves as a messenger between DNA and building the protein. That's one of its main jobs, but there are so many different jobs that RNA does. Honestly, RNA is probably the most interesting chemical, but we're not going to spend much time on it at all. Um, and again, this forms monomer polymer. You get a simple subunit and it links up and becomes long chains. So this is showing the subunit. It is a sugar. Off to the side, there is this nitrogen group, this orange circle. And off of the sugar, the other side of it, there's a phosphate group. And what happens is here you can see this chain. Um, you have this green sugar with its phosphate. That phosphate connects to the next sugar. That sugar's phosphate connects on and on and on. That forms the backbone. And then off to the side are all these nitrogen groups sort of sticking out. Those nitrogen groups are what carry the code of DNA. Again, we're going to do a lot more of this in chapter 16. So definitely know this. Keep it in mind. We're going to learn it again. Here are those letters, um, what the sugars look like. And here's the difference between DNA and RNA. The sugar is slightly different. You can see this extra oxygen shown in red here. Um, that's it. That's really the only difference between DNA and RNA. At a basic level is just that one extra oxygen. So again, DNA is stored in the nucleus of your cell. An RNA copy is made of a gene. That copy of the gene leaves the nucleus of the cell, comes over here. There's this structure called a ribosome. It reads the RNA, and that's how it builds the polypeptide, which becomes a protein. So it seems like a lot, but it's just DNA, RNA, protein. Um, and a real world example of this, like an an mRNA vaccine, like was developed during COVID. Um, Basically, it's just this little strand of a message. That message gets delivered to your cells. Your cell just reads it and builds a protein, and that protein becomes the antibody that your immune system can recognize. So basically, it's just saying, hey, cells, make this protein, and then your body recognizes that protein as a bad guy, as an invader. Um, and we can make these mRNA strands really quickly and inexpensively now, based on this new mRNA technology. Also, this doesn't work in reverse. You can't go from protein back to mRNA, back to the nucleus. These messenger RNAs only can leave the nucleus. So even if you get an mRNA vaccine, that can't get into the nucleus. It doesn't go in, it can only come out. So it'll never alter your DNA. The next group of biological molecules are carbs. Um, Carbs serve two major functions in living things. They're either fuel to help run your cells, or they're building materials to give an organism its structure. Um, some of them are simple sugars like fructose or glucose, um, and then some of them are large polymers like starch. The chemical makeup of this is for every one carbon, there are two hydrogens and one oxygen. So, for example, glucose has the chemical formula of C6H12O6. So it's in this 1 to 2 to 1 ratio. Glucose just happens to have six carbons, 12 hydrogens, six oxygens. And again, this is monomer polymer chemistry. Um, the monomer is a monosaccharide, which just means single sugar. Two of those get linked together to make a disaccharide, two sugars, and then a bunch get linked together to make a polysaccharide. It can be three, it can be 100, it can be, 1,000. So you're just linking chains together. In plants, plants are going to store energy in the form of starch. So that's why potatoes are so rich in starch. It's the plant storing energy for itself. In animals, we store carbohydrates in the form of glycogen. Glycogen is very, very similar to starch, but we store it in our muscles and in our livers. But it's basically just animal starch. And then in some organisms, these become structure. They give something its physical structure. In plants, the primary thing is cellulose. That's what gives stems like their rigidity, and leaves are flexible yet strong. That's cellulose. And in bugs and other arthropods like lobsters, their exoskeleton is made um, of the carbohydrate chitin. So normally we depict a sugar in this ring structure. Okay, so we could also draw it in a linear structure, but the way that sugar behaves when it comes into contact with water is it forms a ring. Um, so occurring in your body, it's always going to be this ring structure. But sometimes we abbreviate it in this linear structure so we can sort of see what's going on a little bit. And again, it's just monomer polymer chemistry. You take an OH off of one side and H off the other side, dehydration, synthesis. You then join the two sugars together. And then the reverse is true um, for building sugars up. Okay, so in living cells, here is what starch can look like. There's several different types of starch, but basically it's all just glucose strung together in these long, 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 long chains. Some of the starch is called amylose, and you can see here it's sort of this straight unbranching chain that sort of spirals around. There's also amylopectin or amylopectin, which has a little bit of branching structure to it. But again, it's just all glucose, 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 all just linked together. In our cells, if you eat starch, your body breaks off all those glucoses and then reassembles them into this structure called glycogen, which is a much more branched um, carbohydrate. So you sort of see the difference in structure between the types of starches and the way that our bodies store sugars in this glycogen molecule. In cellulose, again, it's just a different type of glucose. It's still glucose, but it's a slightly different version. It's just glucose, 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 long, long, long strings of them. And they're going to hydrogen bond to each other and form these bundles of threads, basically. And the plant cells are going to weave these threads into this mat. And that cellulose mat is what gives plants their strength and rigidity. Chitin, again, is a slightly different carbohydrate that is used for insect exoskeletons. The last category of biological molecules is lipids. Lipids are a little bit different. They don't follow the monomer polymer trend exactly. Um, they sort of do, but not exactly. The most common type of lipid that you're going to come in contact with is what's called a triglyceride. Um, so I'll just show you on the next slide. Here in this sort of tan box is a glycerol molecule, and then attached to each one of these carbons of the glycerol is a long, long, long fat tail. So it's a glycerol with three fats attached to it. Um, that's triglyceride. And that's the stuff that your fat cells store. As you get older, you have to get blood tests done to see if there's this stuff just floating around freely in your blood. If you have high triglycerides, it can lead to cardiovascular disease and things like that. There's really cool chemistry for how your body deals with fat as it enters your bloodstream and goes to your liver and gets processed. There's really cool stuff that happens, but that's well beyond what we're going to talk about. Another group of lipids, totally different from the triglycerides, are steroids. These are ring structures. And the thing that sort of define something as a lipid is it's a hydrocarbon. It's basically all carbons and hydrogens and no other uh, types of elements. There's no oxygen, no nitrogen, no sulfur, none of that stuff. Lipids are just carbons and hydrogens. So steroids are just rings of carbons and hydrogens. Um, Cholesterol is probably the most common steroid found in animals, but for us, your body's going to change the structure of cholesterol and turn it into estrogen, and then estrogen gets modified into testosterone. So, depending on how your body processes cholesterol into which hormones, um, gives you a lot of your secondary sexual characteristics. And then the third major type of lipid are called phospholipids. And I'll show you that in a minute. Okay. So before we talk about the phospholipids, we need to talk about the different types of fats that we eat. So here on this molecule at the top, you can see every one of these carbons is connected to another carbon with a straight single bond. That is called a saturated fat. And the reason it's called saturated is because every single carbon bond that's available is filled up with a hydrogen. There's no empty bonds anywhere that you could possibly fit another hydrogen into. So it's saturated. Some triglycerides between the carbons will have double bonds. Those double bonds basically are going to remove two hydrogens for every single double bond because it needs those electrons to make the double bond. And that is called an unsaturated fat because it's not totally covered in hydrogens. So most animal fats are saturated fats, and most plant fats are unsaturated. There are exceptions. Um, an example of a saturated fat from plants is coconut oil. Your body metabolizes that really well. It doesn't just store it and do bad stuff with it. Um, and then an unsaturated fat in animals would be something like fish oil, another really healthy oil to eat. So again, here is that ring structure of cholesterol. You can see it's nothing like the triglyceride. And again, this gets modified to get turned into estradiol and then eventually testosterone. But this is the basic steroid that mammal bodies make, at least. And then here is that last one, a phospholipid. So this looks a little bit more complicated, but it's really not. It's just a triglyceride. So if you look right here, these three carbons, that's your glycerol. You have a long saturated fat tail. Here you have one unsaturated fat. See the double bond. And then off of the third carbon of the glycerol is just a phosphate. So that's what those red things are in that chemical. Um, and this isn't always consistent. Sometimes there's more double bonds uh, in those fat tails. There's some variability there, but they all share one thing in common. This phosphorus head is really hydrophilic, loves water. These fat tails are really hydrophobic. So it's a molecule that half of it loves water and half of it hates water. Because of that, this is what forms the membranes of our cells and in our cells. It makes a really good barrier that water can go up against the phosphorus side, but then not pass through the fat side. We'll talk a lot more about this in chapter 7. So keep this one in mind. It's really important. And then this table at the end of the chapter just summarizes most of what we've just talked about really nicely. Um, talks about what what are the four things, examples of the four different molecules, and then what do they do in your body. So that's basically it for this chapter. Really spend a lot of time understanding the protein folding part of it, um, and just knowing examples of all of these things. So that's it.